Very good morning, students. In the present video lecture, we shall learn something about the mechanism of photosynthesis under Unit 3 of Paper 7, Plant Physiology and Metabolism. See, photosynthesis is a plant biochemical reaction wherein both reduction and oxidation reactions occur. Hence, photosynthesis is also referred as redox process, wherein during photosynthesis, water is oxidized, one of the raw material, and another raw material, carbon dioxide, gets reduced to form carbohydrate. We have two reactants in the photosynthesis, both are inorganic, water and carbon dioxide. One of the reactant, water, gets oxidized and another reactant, carbon dioxide, gets reduced. Since both oxidation reduction phenomena takes place during the mechanism, photosynthesis also referred as redox process. Redox process. See, to define photosynthesis, photosynthesis can be defined as a complex organic synthesis of a complex organic food substance like carbohydrate or a glucose from two simpler inorganic substances, that is raw materials like water and carbon dioxide, in presence of solar energy in the green cells of the plant body containing chlorophyll pigment, with the release of oxygen as a byproduct. This overall process is referred as a photosynthesis. See, I'll show the reaction here. See here, the sugar, that is carbohydrate, is synthesized here. So there is a sunlight, solar energy, and the reaction takes place in the green cells containing chlorophyll pigment, and there are two reactants. Both the reactants are inorganic in nature and they are simple when compared to the product. So let us define this. So synthesis of a complex organic substance like a carbohydrate, that is sugar, from two simpler inorganic substances like water and carbon dioxide, in presence of solar energy in the green cells containing a chlorophyll pigment, with the release of oxygen as a byproduct at the end of the reaction. This overall process is referred as a photosynthetic phenomena. Say, Blackman in the year 1905 pointed out that the process of photosynthesis consists of two main phases. That is, the mechanism includes two main phases. The first phase is called as a light dependent phase called light reaction, and the second one is called as a light independent phase called as a dark reaction. Dark reaction. So, light reaction, otherwise called as a light dependent reaction, was first discovered by Robert Hill. Hence, it is also called as a Hill's reaction. Hill's reaction, where during this it is also called as a photochemical reaction. It is because this takes place in presence of photo, that is light, and the chemicals are produced. Hence, it is called as a photochemical reaction. See here, the light reaction takes place in the grana part of the chloroplast, that is in the membranes of the thylakoids, where the photosynthetic unit 1 and 2 are present. That is the site of light reaction. See, light dependent phase or a photochemical phase, during light reaction, oxygen is evolved and assimilatory products like ATP, adenosine triphosphate, and NADPH2, nicotinamide adenine dinucleotide phosphate, reduced are formed. These two are formed during light reaction. Whereas, on the other hand, a dark reaction, otherwise called as a light independent reaction, this was first discovered by Blackman, hence it is also called as Blackman's reaction. Blackman's reaction. See here, during this Blackman's reaction, the assimilatory power which is formed during light reaction, that is ATP and NADPH2, are utilized here for the synthesis of a glucose. So that is why the dark reaction indirectly depends upon light reaction, but independent of light. That is to be noted. Oxygenic photosynthesis, that is photosynthesis which evolves oxygen at the end, is called as oxygenic photosynthesis, which takes place in all eukaryotic higher plants and also in cyanobacteria, that is blue green algae. Whereas non-oxygenic, that means to say without the evolution of oxygen, sometimes photosynthesis takes place, anoxygenic or non-oxygenic, and this takes place in photosynthetic bacteria, for example, where there is no liberation of oxygen. So the light independent reactions, that is light reactions, photosynthesis in the leaves of the plants involve many steps. That is, it can be divided into two stages. That is, as I told earlier in the last slide, independent reaction, light reaction, and light independent reaction, dark reaction. Why this dark reaction is called Calvin cycle? This is because the different reactions of the dark reaction takes place in a cyclic form, and that was first discovered by Melvin Kelvin. Hence, dark reaction is also called as a Calvin cycle. The light dependent reactions takes place in the thylakoids. I told you earlier already, thylakoid membranes, and require a continuous supply of light energy for light reaction to happen. And the light energy absorbed by chlorophylls, that is chlorophylls means to say they are present in the photosynthetic units. In the previous lecture we have learned there are two photosynthetic units, photosynthetic system unit 1 and photosystem unit 2, where photosystem unit 1 has got 250 pigment molecules, photosystem 2 has got 300 pigment molecules, where all pigments absorbs light energy and it is converted into a chemical energy through the formation of two compounds, an energy storage molecule and reduced electron carrier. That is why the light energy absorbed by the pigment molecules in the membranes of the thylakoid is converted into a chemical energy. Hence, light reaction is called as a photochemical reaction. That is, chemical is produced during light. Hence, it is called as a photochemical reaction, which was discovered by Robert Hill. In this process, water molecules are also converted into oxygen gas, and that is the oxygen we breathe actually. That is, photolysis of oxygen also takes place. What is photolysis? Splitting up of water molecule into hydrogen and hydroxyl ions, H plus and OH minus ions. The OH minus ions thus formed recombines together to form water and oxygen, and that oxygen is produced as a byproduct of photosynthesis, which comes from the water and not from carbon dioxide, though the carbon dioxide contain oxygen. The Calvin cycle, also called the light independent reaction, I told you already. It takes place in the stroma region of the chloroplasts and not directly require light. That is why it is called dark reaction. In the grana region of the thylakoid, the light reaction takes place. In the stroma region, the dark reaction takes place. Instead, it requires a stored light energy, indirect form of light energy, in the form of chemicals such as NADPH, and whatever the light energy which is absorbed by the chlorophyll pigment molecules is get converted into chemical energy, and that chemical energy is stored in reduced substances like NADPH. During dark reaction, the indirect form of light which is present in these substances will be utilized. That is why it is called as light independent reactions. What for it is used? To fix the carbon dioxide and produce three carbon sugars, namely 3-PGA, that is phosphoglyceric acid. This is the schematic representation of light independent reaction, that is light reaction on the left side of the diagram, and light dependent, and on the right side we have light independent, that is dark reaction or Calvin cycle. You can see here in the previous slide I told that the light reaction takes light independent reaction, dependent reaction, light reaction Hill reaction takes place in the grana region. What is grana? Grana is nothing but a structure which is present in the stroma of the chloroplast, wherein number of plate-like structures are piled one above the other. These individual plate-like structures are called thylakoids, and on the membranes of the thylakoids, the photosynthetic units, that is quantasomes, are present, and in that where actually the light reaction takes place. So here it is granum. Light always travels from the solar sunlight in the form of photons, that is bundle of light rays. The amount of light energy which is stored in these photons is called as a quantum. So the light energy when hits the chlorophyll molecule, all the pigments in the here photosystem, 250 pigments and 300 pigments in photosystem 1 and 2 respectively, all of them absorb light energy, and that light energy is converted into chemical energy, where during that assimilatory powers, in the previous slide we learned assimilatory powers like ATP and NADPH2 NADPH are formed during light reaction. These ATP, the light which is absorbed by these photosystems or quantasomes or pigment molecules is now stored in ATP and NADPH, that is indirect form of light. These assimilatory powers enters this Calvin cycle or light independent reaction or dark reaction, where when they enter, the energy, light energy stored in these assimilatory substances is used by this Calvin cycle again to wherein during Calvin cycle carbon dioxide is absorbed by the chlorophyll pigment molecule, chlorophyll pigments. So this carbon dioxide enters where the in the previous slide we learned that 3-PGI is formed as the stable product, phosphoglyceric acid, and again ultimately it releases the NADP, NADPH, where H is utilized here. In ATP, one of the phosphate is utilized here. So first ADP is released and NADP is released. Again, this NADP and ADP accepted by this grana again to form assimilatory powers. This is how, though the light reaction and dark reaction, light dependent and light independent, both takes place inside the chloroplast. They are related to each other, but the mechanism here is different one in during light reaction, and mechanism here is a different one in presence of separate enzymes, biocatalysts. This is what written here. The light dependent reaction takes place in the thylakoid membrane, that is this one, light reaction. They require light, and their net effect is to convert water molecules into oxygen. This is the oxygen which is released here during photolysis of water, while producing ATP molecules from ADP and IP, and NADPH molecules, reduction of NADP, that is NADP is formed. ATP and NADPH are produced on the stroma side of the thylakoid membrane, where they can be used by the Calvin cycle. You know this already. The Calvin cycle takes place in the stroma region of the chloroplast, where the enzymes are located for required for dark reaction. It uses the ATP and NADPH from the light dependent reaction. These two, when ATP and NADPH utilized by this Calvin cycle, what for they are utilized? To fix the carbon dioxide. Here is the atmospheric carbon dioxide which enters the Calvin cycle. To fix that carbon dioxide into a carbohydrate, these two are utilized. The Calvin cycle converts ATP to ADP. Here it is ATP, it absorbs ATP and releases ADP. You can see here. And NADPH to NADP. See, it absorbs NADPH and converts it into NADP. The ADP and IP, NADP can be reused as substrates in the light reaction. I told you already, these two are accepted by the grana part of the chloroplast again to continue the cycle. This is how the in the same structure, in the same organelle, that is chloroplast, both light dependent and light independent reactions takes place. Moving on to the quantum requirement and quantum yield. What is that quantum? The solar light comes to the earth in the form of small packets of energy known as photons. I told you in the previous slide, the light energy from the sun always travels in the form of bundles, and the bundle of light rays is called as a photon. In that photon, the energy associated with each photon is called as a quantum. The amount of energy present in each photon is what is called as a quantum. What is this quantum yield then? See, the number of photons are quanta required by a plant body or a leaf to release one molecule of oxygen during photosynthesis is called as quantum requirement. That means to say that during photosynthesis, inorganic raw materials like water and carbon dioxide, they react together to form carbohydrate and they release oxygen. So to release one molecule of oxygen as a byproduct, what is the amount of light energy the leaf utilized requires or the plant requires is what is called as a quantum yield. Quantum yield means to say the amount of light energy required by the plant body to release one molecule of oxygen during photosynthetic reaction is what is called as a quantum this is what is called as a quantum requirement. It has been observed that in most of the cases the quantum requirement is 8. It means that 8 photons are required to release one molecule of oxygen. To release one molecule of oxygen, the number of quanta light required are 8. The number of oxygen molecules released per photon of the light during photosynthesis is called as quantum yield, already I defined. If the quantum requirement is 8, then the quantum yield will be 1 by 8. That is simple mathematical calculation. So here the photosynthetic units, otherwise they are called as quantasomes, we have learnt already in the previous lecture. It is defined as a smollett group of collaborating pigment molecules necessary to bring about a photochemical act, that is light reaction, that is absorption and migration of a light quanta to trapping center, that is reaction center P680 or P700 accordingly, where it promotes the release of an electron. Here comes to picture the two scientists, Robert Emerson and Arnold. Emerson and Arnold, 1990 or 1932, on the basis of the experiments, stated that about 250 chlorophyll molecules are required to fix one molecule of carbon dioxide in the atmosphere. See, the reaction says that 6 molecules of water combines with 6 molecules of carbon dioxide to form a glucose molecule. To fix one molecule of carbon dioxide, how much of chlorophyll pigments are required according to Emerson? It is 250 pigment molecules. So 250 into 6, 1500 chlorophyll molecules are required to fix 6 molecules of carbon dioxide as per the reaction. The number of chlorophyll molecules was called the chlorophyll unit, but the name was subsequently changed to photosynthetic unit, and later it was designated as a quantasome by the scientists Park and Begins in the year 1964. Even this is also dealt in the previous lecture. The size of the quantasome is about very meager size present in the thylakoids. Chloroplast itself is microscopic. In that chloroplast we have grana. In that grana we have thylakoids. So thylakoid contain the quantasomes. So just imagine the size of the quantasome, very minute. Each quantasome consists of about 200 to 240 chlorophylls, again 48 carotenoids, 46 quinones, 116 phospholipids, 144 diglycerides, 346 monogalactosyl diglycerides, 48 sulfolipids, some sterols, and special chlorophyll molecules, that is reaction centers P680 and P700 accordingly. That is the composition of a quantasome. P is the pigment 680 or pigment 700 donates the wavelength of light these molecules absorbs for the conversion of light energy into chemical energy, that is reaction center. P680 or 700 constitute the reaction center of the photocenter. Other accessory pigments and chlorophyll molecules are to light energy or antenna molecules. It captures sunlight and transfers it to the reaction center by resonance transfer or inductive resonance mechanism. So here is a phenomena called as red drop and Emerson effect. What is this red drop? Red drop means to say that Emerson and Lewis, these are the two scientists who conducted experiments on photosynthesis in a plant called chlorella belonging to a group called alga. Chlorella is an algal member. So on that plant, when they conducted photosynthetic experiments, there was a sudden drop in the rate of photosynthesis. You look at this graph. There is a sudden graph. This is the wavelength. This is x-axis shows the wavelength. There is a sudden drop in the at 680. See, it has started with 660 and it is 680. There is sudden drop in the rate of photosynthesis. 680 is a red region, at the red region of the spectrum. This sudden drop in the rate of photosynthesis beyond the red region of the spectrum is called as a red drop. Is called as a red drop. Drop in the photosynthetic yield beyond 680 nanometers of the wavelength is called as red drop, according to Emerson. This decrease in quantum yield took place in the far red part of the spectrum, that is, the curve shows the quantum yield drops in the region about 680 nanometers. This is region right side to it above and left side to it is below. So there is a drop, sudden drop. This is drop in the rate of photosynthesis at the red region is called as red drop. This decline in the rate of photosynthesis itself is called red drop effect. Later, Emerson and his co-workers like Lewis, they realized that the inefficient far red light in chlorella beyond 680 nanometers could be made fully efficient if they supplemented with the light of a shorter wavelength. If the same chlorophyll is subjected to an alternate light of short and wavelength and a longer wavelength. Alternatively, then there was a sudden increase in the rate of photosynthesis. Look at the graph, which is mentioned, which is written in the dotted line. So sudden increase, enhancement in the rate of photosynthesis when this chlorophyll is subjected to light of shorter wavelength and longer wavelength simultaneously and alternately, there is an increase, enhancement in the rate of photosynthesis. This enhancement itself is called as this enhancement effect. This enhancement of photosynthesis itself is called as Emerson enhancement effect. So there is a drop in the rate of photosynthesis at beyond 680 nanometers. This is called as red drop. To balance this, when the same chlorophyll is subjected to an light of shorter and a longer wavelength simultaneously, then there was sudden increase, enhancement in the rate of photosynthesis. This itself is called as enhancement effect. Enhancement effect. The same thing is being explained here. The same graph which was in the previous slide is explained here. Emerson and Lewis, while determining the quantum yield of the photosynthesis in chlorella by using monochromatic light of the different wavelengths, noticed a sharp decline in the quantum yield and at 680 nanometers. This decrease in quantum yield is what is called as a red drop. This Emerson and his co-workers in 1957 found that the inefficient for the red light of the chlorella beyond 680 nanometers could be made fully efficient if supplemented with light and short wavelength alternatively. The quantum yield from the two combined beams was found to be greater than the effect of both beams when used separately. This enhancement of photosynthesis is called as Emerson effect. By knowing the preliminary mechanism of the photosynthesis, that is the first part of the photosynthetic mechanism, light dependent light reaction, which takes place in the grana part, and light independent dark reaction takes place in the stroma region of the chloroplast, and how they are interconnected with each other, and what is red drop, and how the sudden drop in the rate of photosynthesis increased in Emerson effect. By knowing all these things, in the next lecture, we can move on to the mechanism of light reaction. Thank you.